So I'll be talking about Rhabdoviruses, their importance, diseases they cause, and before I do that, I just wanted to provide a bit of context. If I can get this to work. There we go. Obviously, food security, particularly in relation to our growing world population, is a very important issue that concerns all of us, and that is important for both food quantity as well as quality. Plant pathogens contribute, according to this slide, about 13% of losses to production and quality of many of our fruits, vegetables, and broadacre crops. So they are an important part of the production system to be analyzed. And I'll be particularly concentrating on viruses, and you can see from this slide there's a huge variety of different shapes and forms and genomes in these viruses. And this is reflected again here in this virus sphere. And on this slide, you can see the different genome types of viruses shown as DNA or RNA. And there's also the different colors that indicate what types of hosts these viruses infect. So green represents plants as hosts, red is vertebrates, brown is invertebrates, and in here we've got some fungal viruses. The viruses I'd like to talk to you about this afternoon are these single-stranded negative sense RNA viruses in this blue sector. What is a negative sense RNA virus compared to a positive sense RNA virus? A negative sense RNA virus needs to make a messenger RNA before it can translate its protein, whereas a positive sense RNA virus, the genome is the same as the messenger RNA. So when a positive sense virus infects a plant, it can immediately make proteins and start its replication cycle. Whereas a negative sense RNA virus is a little more complicated in that it needs to bring proteins with it to make transcripts of all its genes before it can get going and make further proteins. So these viruses are rather difficult to study, even though they've been quite successful in the environment to infect various organisms. And that includes... These viruses have a very diverse host range, and you can see this when you look at the colors in that blue segment. There's green for plants, invertebrates, vertebrates in red. And one subsection of these viruses that I'll be talking about are mononegaviruses. These viruses have their whole genomic information in one molecule, in one piece of RNA. And in particular, I'll be talking about these things here, which you see in all three sectors of the single-stranded negative sense RNA viruses, which have a bullet-shaped or bacilliform morphology, and they are called Rhabdoviruses. So this is the overall taxonomy of the mononegavirales or mononegaviruses, the ones with that one molecule of negative sense RNA. And you will see on the top there's the Paramyxoviridae, which is one of the families in this order, and the probably most well-known virus for Queenslanders in this is the Rhabdovirus. So a very important mononegavirus. Just below is the Filoviridae, and Ebola is part of this. Then we've got the Niamiviridae. They're a new family of viruses that infect ticks and seabirds. And the viruses that I'll be talking about are in this other family with the red ring around it, the Rhabdoviridae, the Rhabdoviruses. So you'll see in this sort of somewhat enlarged area of the Rhabdoviruses, there's a group down the bottom, this where you see the rabies virus as one of the representatives. This is all the animal infecting Rhabdoviruses. And then on the upper branch here, thanks, these are the plant-infecting Rhabdoviruses, and they fall into two genera, Cytorhabdovirus, because they assemble in the cytoplasm, and Nucleorhabdovirus, because they replicate in the nucleus of infected cells. So I'll do some in-presentation blatant advertising here as well. I've co-edited with Ivan Kuzmin from the CDC a book on Rhabdoviruses in 2012. But the reason I'm showing this is for the shape of these viruses and where the name comes from. It comes from the ancient Greek rhabdos, meaning rod or wand. And these viruses infect a very large diversity of organisms. They infect fish, they infect bats, they infect land mammals, plants, insects, and they have slightly different shape in that the ones that infect animals are usually bullet-shaped, so they have one flat end and a round end, whereas the plant-infecting ones generally have a bacilliform shape, so they are round at both ends. And they cause quite a number of important diseases. For example, here in fish, viral hemorrhagic septicemia, a very important disease. These viruses in fish are transmitted by water. So the fish can pick them up just by swimming in infected water. Another important disease, bovine ephemeral fever. It's another rhabdovirus that infects cattle. And rabies, again, a large host range for a rhabdovirus infecting dogs and also being able to infect humans. So on the plant side of things, where I'm located, there are also a number of diseases caused by rhabdoviruses, and the plant rhabdoviruses, at least all the ones where this has been studied, are transmitted by insects. They're transmitted either by aphids, like lettuce necrotic yellows virus, and I'll be talking a bit more about this Australia-New Zealand virus later. Maize mosaic virus is transmitted by plant hopper, Peregrinus maydus. And there's a few examples of agriculturally important viruses, like the one causing Alomi disease in the Pacific Islands in taro, alfalfa dwarf, a newly recognized virus in alfalfa in South America, and potato yellow dwarf. This is a healthy potato, and these are infected potato plants here. So lettuce necrotic yellows virus, native Australian virus. We don't really know how and when it arrived, but at this stage it occurs only in Australia and New Zealand. And it has a interesting epidemiology in that its main host of the virus is a weed, sow thistle. And the main host of the insect is also the thistle. The virus replicates in the plant in the thistle, but it also replicates in the insect. So this virus has two hosts, an animal host and a plant host. The economically important host, like lettuce or chickpeas or garlic, so this virus has been pulled out of quite a few different species, reaches these hosts by the vector feeding, and because the vector is infected for life once it has taken up the virus, and the virus keeps multiplying in the insect, wherever the insect lands and feeds, it deposits the virus, and in most cases the plants can get infected. So the host range is mostly determined by where the insect lands rather than by the virus per se. These pictures are taken in the Lockyer Valley. You can see these plants, they were all planted at the same time. These were infected very young and they don't develop any further. And these ones have been infected at a later stage. This is a late infection, again in the Lockyer Valley with Dennis Pursley standing here. The plants get, because the leaves all fall down on the ground, they get secondary fungal and bacterial infections. And so this is a total write-off. Now, Rhabdovirus particles, when you look at them more closely, they contain 5 different proteins. There's the RNA, negative sense RNA, which is very tightly associated with this so-called N protein, which is a nucleoprotein or nucleocapsid protein. There's 2 other proteins that are essential for the virus to start replicating. One is the L protein polymerase, and the second one is a phosphoprotein, which is a cofactor. And these 3 proteins, together with the RNA, form what's called the nucleocapsid core, and that is the infectious unit of the virus. Positive sense RNA viruses, the RNA is infectious by itself. You don't need any proteins. But negative sense RNA viruses, you need these 3 proteins in order to cause an infection together with the genomic RNA. And that's what makes it so difficult to study, because we don't really, particularly not for plant Rhabdovirus, we don't have any infectious clones. So we need to resort to other methods to study these viruses. So in addition to the nucleocapsid core, we have a glycoprotein, which sits in a lipid membrane, which the virus picks up from its various hosts, the insect or the plant. And this is all connected together by this matrix protein, which connects the glycoprotein, the internal part of the glycoprotein, to the nucleocapsid core. I mentioned that the transcripts need to be made. So this is the genome, 3' to 5', with our 5 proteins, N, P, M, G, and the L polymerase. And these get sequentially transcribed to make the messenger RNAs and nanoproteins. Now, one of the studies we've recently expanded was to look at the genetic variability of this virus. Knowing that it occurs in Australia and New Zealand, we've collected samples from both countries, and we've done a variability study using the entire nucleocapsid gene sequence. And when you do this and put it into a phylogenetic analysis program, you find 2 distinct subgroups. And members of both subgroups, here's an AU and an NZ, and the same here, occur in both countries. However, subgroup 1 was last seen in Australia more than 15 years ago. It has disappeared, whereas it's still present in New Zealand. And you can see an example here. The yellow isolates on the Australian map are subgroup 1 isolates, whereas the blue ones are subgroup 2. And in 1993, both isolates occurred in the same area around Adelaide. But subgroup 1, as I said, has now disappeared. We don't know why this is. It could well be that one group of isolates is more easily transmitted by the aphids than the other one is. and this change still seems to be happening in New Zealand, so it'll be very interesting to follow this up across the ditch. Because lettuce necrotic yellows virus, even though it has lettuce in its name, it's very difficult to study in this host because it is not mechanically transmissible. And so we're using a Nicotiana species, Nicotiana glutinosa, in order to study this virus because we can mechanically rub inoculate the virus onto the leaves. And when we do this and look after 2 weeks, we can see that different isolates cause very different symptoms. This is a very severe isolate, causes necrosis on the inoculated leaves, and then a mosaic and a leaf curling on the systemic leaves. And this is a New Zealand isolate, which is quite severe, whereas this one is a very mild isolate. And this one, you can hardly see any symptoms, even though there's just as much virus in this plant as in this plant. Now, looking at the genome of Rhabdoviruses across the board, and here again is those 5 genes, N, P, M, G, and L. They are accessory genes that the viruses have incorporated into this generic genome structure. And in most cases, these accessory genes have inserted between the existing open reading frames rather than within a gene which might interrupt the function of the gene. And so there's genes inserted in this junction between the N and P genes, P and M, G and L, and in most cases there's a gene inserted between P and M. And in particular, in plant Rhabdoviruses, we have a gene here, inserted here that assists in the cell-to-cell movement of viruses through the plasmodesmata. And this is seen here. This on the top, you have the genome organization of VSV. Animal Rhabdovirus, whereas here between P and M for these 3 viruses, this is a cytorhabdovirus, LNYV, and 2 nucleorhabdoviruses, we have a movement protein gene inserted. It's got different names in different viruses, but it appears to have the same function. I mentioned that there's 2 genera. The nucleorhabdoviruses arrange a virus factory production in the nucleus of infected cells, whereas the cytorhabdoviruses, their viral plasm is in the cytoplasm of infected cells. And this obviously causes quite significant differences between these two types of plant Rhabdoviruses. I should mention that most plant viruses and most viruses seem to replicate in the cytoplasm of infected cells and not in the nucleus. This is the only group of Rhabdoviruses that replicates in the nucleus. All other Rhabdoviruses are cytoplasmic. So when a virus enters the cell, it associates with ER membranes, establishes a viroplasm, makes messenger RNAs, translates those into proteins, and particles get assembled and bud through ER membranes where they pick up their glycoproteins, and they also move cell to cell. So it's pretty straightforward for a cytorhabdovirus. For nuclear rhabdoviruses, here on the right-hand side, you see a similar picture after entry into the cell, association with membranes, but then the genome and proteins have to be imported through the nuclear pore complex into the nucleus of the cell, where this viroplasm is established. In order for the mRNAs to be translated, they have to be transported back out into the cytoplasm. and then the proteins have to be re-imported into the nucleus in order to produce the virus particles. So there's a lot of interactions going on in and around the nucleus for the replication of this group of viruses. Now I want to change tack a little bit and talk about protein-protein interactions and their importance. So when you're thinking of it, viral proteins need to interact in order to form virus particles so that they can move and be transmitted. But they're also required for the infection itself, for replication, for, as I mentioned, the virion assembly, for movement, and also there are interactions for defense responses. So there's interactions amongst the viral proteins and between the viral proteins and the host proteins. And we've been studying this in my lab using Agrobacterium tumefaciens infiltration into Nicotiana benthamiana leaves, this little native Australian plant. So we engineer our protein expression constructs as fusions with various autofluorescent proteins and then visualize them in confocal microscopy. And we use different colors that are well separated spectrally, like we're using green versus red and cyan versus yellow. And you will see this in some examples. Another tool we're using are transgenic Nicotiana benthamiana plants where some organelles of the plant are fluorescently labeled in every cell. And we've obtained those from Michael Gooden at the University of Kentucky. So he has generated plants that have the endoplasmic reticulum fluorescently labeled or the nucleus fluorescently labeled. Again, a reminder of the virus particle. RNA, nucleoprotein, always associated with it, and then the phosphoprotein, the polymerase, the matrix protein, and the glycoprotein. So if we express these various viral proteins in these transgenic plants, this is what we see. This top panel here are green fluorescent protein fusions of our viral proteins. The middle panel is the transgenic plant where the histone 2b protein in the nucleus is fused with red fluorescent protein. So we see red nuclei, which is a great help when you want to focus when you're looking at sections of live cell leaves. And then this is the overlay of the green channel and the red channel. So this first panel here is free GFP. And free GFP localizes to the nucleus and it localizes to the cell periphery. The cell periphery for these type of epidermal leaf cells looks like pieces of a jigsaw puzzle. And because there's a large vacuole in plant cells, the cell periphery is where the cytoplasm is located. So if we start with the nucleoprotein, remembering this is a cyto-rhabdovirus, the nucleoprotein is located in the cell periphery, in the cytoplasm. And here you see your nucleus. This is the phosphoprotein, is also cytoplasmic. The movement protein in this virus called 4b is in the cytoplasm and in the nucleus. And the matrix protein is also in the cytoplasm and the nucleus. Whereas the glycoprotein, as one would expect, is associated with ER membranes all across the cell. This is a repeat of the last slide where you see this is the nucleoprotein in the cytoplasm, the phosphoprotein in the cytoplasm. If you co-express these two in the same cell, you just mix your agrobacterium cultures for your two proteins and infiltrate them into the same leaf, you see a total change. So there seems to be a interaction happening and a relocalization of N and P together somewhere near but outside of the nucleus. So to test for this, that this is a real interaction rather than just a co-localization, we've used another method called bimolecular fluorescence complementation. For this method, you use a yellow fluorescent protein which is split in two halves, and you fuse one protein to one half and another protein to the other half. If these two proteins, A and C, do not interact, you don't see any fluorescence. If the proteins A and B do interact, they bring the two halves of the YFP together, so it reconstitutes some of the YFP, and you can visualize this by confocal microscopy. And this is what this looks like. Yellow and cyan are well separated, so that's why we are using a different transgenic line which has cyan fluorescing nuclei with a CFP H2B protein. So here you see four interactions. The P protein interacts with itself, the N protein interacts with P, and so this looks very similar to what we've seen in the co-localization. So this is a true interaction between the two proteins, because otherwise you would not see any YFP. The matrix protein interacts with itself in the cytoplasm and in the nucleus, and the M protein interacts with the phosphoprotein, looks somewhat similar to the interaction of N and P. So with this method you can see a protein-protein interaction, but you can also see where this interaction happens, so you can localize and look for interaction at the same time. Now, when you compare this across to the nuclear rhabdoviruses, you see some conserved interactions. So this is what we've just seen for lettuce necrotic yellows virus, NP interaction. You see the same NP interaction for nuclear rhabdovirus. The main difference is that you see it in the nucleus rather than in the cytoplasm. And the same is true for the matrix protein self-interaction. Happens in the nucleus, very, very distinct in the nuclear rhabdoviruses, and it happens in the cytoplasm and the nucleus in lettuce necrotic yellows, the cytorhabdovirus. Now, my colleagues at the University of Kentucky, they've taken this to the next step, and they thought, well, we find an interaction between the N and the P protein, which is indicated by this line. We find a self-interaction of the P protein indicated by the circle. Let's see what host proteins interact with these viral proteins. And so they used a yeast-to-hybrid system of a large library from Nicotiana benthamiana, and they identified quite a number of interacting proteins. So this is a N interacting protein number 67. And what this shows is that, for example, the movement protein, which in this case is called SC4, interacts with a interactor, a host protein number 17. But at the same time, the matrix protein also interacts with this same host protein. So this host protein provides a link between those two proteins in helping during the virus replication. Even more complex, this SC4 protein also interacts with this protein number 21, and the nuclear protein interacts with Ni67, and these two host proteins interact with each other, again facilitating an interaction between these two proteins via those two host proteins. So this is a very complicated interactome, you might want to call it, between the viral and the host proteins. And in this Min et al. Publication, they've proposed a model on how Sonka's yellow net, which is a nuclear rhabdovirus, moves, in that it uses all these viral proteins. So there's not a dedicated single movement protein, but this is the movement protein, glycoproteins involved, the matrix protein, the nuclear protein, and these still to be identified host proteins, they all form a movement complex which then moves along the microtubules in the cell to the plasmodesmata and into neighboring cells. So you can learn a lot even if you don't have any infectious clones available to you. I'd now like to show you some data we've recently obtained on a nuclear rhabdovirus. In Australia, it's called Datura yellow vein virus. It infects Datura, it infects tomatoes, and it infects a ornamental plant called Thunbergia alata, or black-eyed Susan. This is a nice flower. It grows in gardens all around Brisbane. And all the ones I've sampled all have got this virus. So it causes these vein yellowing, vein clearing symptoms on the leaves. So this is a nuclear rhabdovirus, established a virus replication factory in the nucleus and has these typical bacilliform rhabdovirus particles. The genome organization we've recently determined, this is the basic structure, and it's just got this movement protein between a P and M, similar to its closest relative, Sonkus yellownet virus I showed you before. This is the percent sequence identity, which is not all that high. The highest percentage identity is with the nucleoprotein. Rhabdoviruses in plants are very much distinct from each other. This, you have to remember, this is the closest relative of this virus. All the proteins, all the genomes are very, very dissimilar to each other. These viruses, I've showed you this table because the nucleoprotein has a predicted very strong nuclear localization signal. The highest score on this is 12. So this is a pretty strong nuclear localization signal, at least predicted by the software. The other high ones is the matrix protein and the polymerase. So this is a nuclear rhabdovirus. You would expect some of these proteins to be present in the nucleus. And this is what we see when we do the fluorescent protein localization. Here's the nucleoprotein of this virus, clearly in the nucleus and a little bit in the cytoplasm, again with a reference to the red RFP histone 2b. The phosphoprotein is in the nucleus and in the cytoplasm. The movement protein, here called P3, has a similar localization. The matrix protein is interesting. It is totally nuclear. And what you can also see is that when you just look at the red channel, there's a big hole in the middle. And so that shows you that the matrix protein has actually displaced the DNA of the nucleus and pushed it to the side to establish itself in the nucleus. And so we imagine that with the interaction between the matrix protein, the phosphoprotein, and the nucleoprotein, this would then establish a viral plasm in the nucleus to enable virus replication in an infected cell. The glycoprotein, again, localizes to ER membranes. We've also looked for NP co-localization, and this does occur where we have a green labeled nucleoprotein and a red labeled phosphoprotein, and they co-localize. We haven't done the biopsy experiment for this yet to confirm the interactions. And I found this on the internet, and I thought because I was going to talk about a different group of viruses next, I'll show you those of rhabdoviruses being fashionable on fingernails and on thongs. So a slight change coming back to these mononegaviruses. Here's our rhabdoviruses, cytorhabdovirus, nuclear rhabdovirus, and up here there's two other viruses, Orchid fleck virus and coffee ringspot virus. And these viruses have been proposed as a new genus called Dicoviruses. they're sitting just off the nuclear Rhabdoviruses here. And this new genus name, Dico, means split into two or split asunder. And so the name was coined because the genome of these viruses is not mono. It is a bipartite. It occurs in two pieces. But otherwise, the same genes are there, and they are very similar. So you can see here, these are all these proposed dichorhaviruses in this clade, which directly links to the clade of all the nuclear Rhabdoviruses. And that's what these are most closely related to, even though their genome looks somewhat different. And this is shown here. Here is a nuclear Rhabdovirus genome. And this is the genome of Orchid fleck virus, dichorhavirus. The polymerase has its own RNA, whereas all the other genes, N, P, 3, M, and G, are located on RNA 1. So this is a bit of a taxonomic conundrum on how to classify this in a mononegavirus group or order, but we're working on that. So there's a group closely related to the nuclear Rhabdoviruses. And these viruses, the dichorhaviruses, they are not transmitted by aphids, plant or leafhoppers. They are vectored by false spider mites of the genus Brevipalpus. And the diseases they cause are somewhat unique in that the virus does not spread systemically. The virus only causes local symptoms. So where the virus is deposited by the false spider mite, and I should mention that these viruses also replicate in their insect vectors. So again, they have two hosts. So this is hibiscus green spot virus. There's coffee ringspot virus in coffee berries, citrus leprosis, and in most cases they cause quite unique lesions on the plants. There are ornamental trees, various orchids, and here lesions on a citrus fruit. Coffee ringspot virus, this is what the particles look like. They look somewhat like Rhabdoviruses, but they do not have a membrane, or at least not a membrane that seems to stick to the virus. They're still associated with membranes in the host, but the particles themselves, when you isolate them, do not have a membrane. And these are the symptoms caused on coffee leaves and fruit. Orchid fleck virus is the best studied of these viruses, causes symptoms on the leaves. You can find ring spots and various types of symptoms. These are the particles here. So they look very similar to those of Rhabdoviruses. The most serious disease caused by dichorhaviruses is citrus leprosis. A huge cost for mite control, as you can see here in Brazil. These photographs are provided by Elliot Kitajima from University of Sao Paulo. And the interesting thing with citrus leprosis is that it is caused by two distinct viruses. It is caused by a cytoplasmic virus, and it's caused by a nuclear virus, and they're both transmitted by these false spider mites. One is a positive sense RNA virus. It's quite widespread and very severe, and the other is caused by this nuclear dichorhavirus, which seems to be geographically somewhat in a different location than is the positive sense RNA virus. So a lot more research is happening on this citrus leprosis. So to finish off, I mentioned the difficulty of taxonomy for this group of bipartite viruses, and so the current idea is that there may have been a mononegavirus ancestor a long time ago in evolution, which gave rise to the non-segmented mononegavirales like the Rhabdoviruses and the segmented viruses like the Dicoviruses. I'd like to finish off with some acknowledgements. I'd like to acknowledge the people in my group, Shivani, Nicholas, Krin, and Fan, who was a project student with us last year. David Innes has significantly contributed to sequencing the Datura yellow vein virus. Colleen Higgins was here on sabbatical from Auckland University of Technology and worked on the variability study of lettuce necrotic yellows. Michael Goodens provided us with his wonderful toolbox of constructs and plants to do these cell localization and interaction studies. Elliot Kitajima from University of Sao Paulo, he came here for the plant virus workshop in August and spent some time with us. And Hideki Kondo from Okayama University, we are collaborating on comparing the proteins of dichorhaviruses and Rhabdoviruses to see, to get more of an evolutionary idea of how they may have evolved. And Jens Kuhn, from the National Institute of Health, he leads the initiative on taxonomic classification of all the different mononegaviruses. Thank you very much, and I'm happy to answer any questions.